Hello, everyone, and welcome to an introduction to the microbiome. I'm Joanne Geer, and I'm the Executive Director of the Biopharma Research Council. We're essentially an association for scientists across all the many silos of biomedical research: industry, academia, nonprofit, government, and supplier labs. We provide programs throughout the year through webinars, through conferences, symposia, and so on. We have a really diverse group in our audience today. I'm very pleased. Many people from pharmaceutical companies, from small startup companies, from academic institutions, and so on, and internationally. So welcome, everyone. These are all the wonderful people you're going to be hearing from. They'll be introduced in turn. It's a great program. We so appreciate all of their participation and all of their institutions supporting this effort. I'm going to share with you a few things that are coming up next Thursday at the same time. We're going to have the beginning of an occasional series we're calling Global Collaboration to Address Ebola. There's so much happening, and personally I wish we had focused this intensively on HIV when it got started, but maybe there's hope because we're learning how to work as a worldwide community against a key threat. In collaboration. So Dan Didier, who's the head of public health at Thermo Fisher Scientific, will be our speaker, and he'll be addressing all the different efforts that they're involved with throughout the world. Coming up in March is Best Practices in Outsourcing, Preclinical and Early Clinical. This is a 3 days, it's spread across 3 days. It's an executive retreat near Princeton that will bring together those who purchase services, in all phases of the preclinical, goes through animal studies, and the translational aspect. There'll be a major emphasis on the impact of bioinformatics throughout this session. And these is our wonderful chairs, and we've just been booking our speakers, so we'll have more information that'll be going out on that very soon. But it's George Karam at Pfizer actually started this six years ago, saying there really needs to be better communication across these different groups. So we're very pleased to have him still involved. Next August, we'll be returning to North Carolina. This is the Triangle Biotech Research Symposium is a one-day program that really mixes up research that's going on in all different directions, again, bringing university and industry people together. We're very excited. The second version of our D2D, Data to Drugs and Diagnostics, will be next fall. We're working on our location, but we have our dates, and that will be in the Northeast Corridor. You can enter questions at any time. We're going to actually address the questions after all of our speakers, and we'll have a really nice open discussion. But you have a question box on the right-hand side of your dashboard, and you can enter at any time. So, first I'm going to introduce you to Howard Young, who is on our advisory board, our scientific advisory board, and who was the first person who brought up the topic of addressing the microbiome some time ago, and has really been a wonderful leader in our organization. Howard is the Deputy Chief at the NCI Laboratory of Experimental Immunology at the NIH. He received his PhD from the Department of Microbiology and Immunology at the University of Washington and did postdoctoral research under Drs. Edward Skolnick and Wade Parks at the National Cancer Institute. He joined the NCI in 1983 and became Deputy Chief of the NCI Laboratory of Experimental Immunology in 2006. His research is focused on the analysis of cytokine gene expression and signaling, with a special emphasis on the molecular characterization of the transcriptional and post-transcriptional regulation of interferon. Howard Young.
Howard, you need to unmute.
Thank you, Joanne. I'm sorry I had the button muted, but I'm very pleased to be involved in this and what we hope to be a series of webinars on the microbiome. This first one will give some overall views and specific topics, as you'll see. And I'm just showing you here a few of the recent publications that have really emphasized the microbiome and how it may be affecting our health and well-being. The role of the microbiome in human health is just beginning to be explored, and part of this is due to the fact that technology has changed. And by this I mean it's now become affordable for many labs across the country to begin to analyze the microbiome, and I have no doubt that in publications they submit, they get questions back from the reviewers on how the microbiome may be affecting their results. And that's the biggest challenge, I think, of research today in the microbiome, to separate the wheat from the chaff, to figure out what is really a cause, in other words, symptoms that may be a direct result of the microbiome versus changes in the microbiome that are a result of the changes in the host. And as we all have read today, there are many possible changes going on. There's been studies showing the microbiome in obese individuals is very different from that in lean individuals, and it affects how they digest their food. There's evidence to suggest that the microbiome may be different in children with autism. That could affect metabolites that appear in the blood, which could affect brain chemistry. There's strong evidence to suggest that the use of antibiotics in this country may affect cancer therapy. It may affect the overall obesity patterns we have in this country. So there are many examples of how the microbiome can be affecting our health and well-being. And with that, I'll turn it over to Winston to introduce our speakers so we can hear about their specialties and how the microbiome may be affecting different aspects of human health and physiology.
Thank you, Howard. So today I'm also very delighted to moderate this short course on the human microbiome, and a very exciting and evolving area of clinical and translational research. So today we have two leading academic researchers and a developer and application scientist from industry presenting today. And just a few housekeeping items before we begin. So the format of today's session is as follows. Each speaker will have about 8 to 12 minutes to present, and that will leave us, a good amount of time for the questions and answer session after the three presenters have presented. So please remember, you can also continue submitting your questions during the presentations by entering your questions in the dashboard to the right of your screen. So we have our first speaker, which is Dr. Kemi Martin. She studies the impact of nutrition on the development of innate immunity in premature infants and its effect on short-term morbidities and long-term outcomes. Dr. Martin, I've worked with her for quite a few years, and she's assistant professor at Harvard Medical School. She's a neonatologist, the associate director for the neonatal intensive care unit, and she also holds a position in the Division of Translational Research at the Beth Israel Deaconess Medical Center. Her previous work had examined, large cohorts of data where she examined nutritional intake of premature infants and their neurodevelopment. Through that work, it became very evident that I guess we're not very good at providing nutrition to babies so that they can achieve optimal growth and neurodevelopment. She's also built a huge repository of premature infant samples that includes blood, IV nutritional intake, breast milk, stool, and tracheal aspirates. And some of our findings revealed that, which you'll hear some today, about fatty acid levels in babies. Bloods are significantly altered from birth levels soon after delivery in the first postnatal week, impacting their risk of developing lung disease and infection. In addition, she's also created quite a few animal models that they can actually monitor and alter the fatty acid delivery. So Dr. Martin will present today. Her title for her presentation, Establishment of Early Microbiome in Neonates, Determinants, Risk, and Therapeutics. And her abstract is actually on the website, so everyone can refer to that. So I'm going to hand it over to Dr. Kemi Martin to present. Please enjoy.
Thank you, Winston, and thank you to the organizers for inviting me to speak briefly about the importance of the maternal fetal microbiome as well as the establishment of the early infant microbiome. So For this segment of the webinar, I will discuss the influences of that early establishment of the microbiome, the maternal-fetal environment, ex utero determinants, and what are the medical consequences when that microbiome is altered, and just take a peek at some of the strategies to protect and restore our microbial diversity. It has been long taught to physicians, and this was part of my training as well, that the in utero fetal environment was sterile. However, recent microbiome studies have challenged that dogma that this environment is sterile. In fact, it appears that placenta does harbor a unique microbiome. In the study referenced here, they demonstrate that the microbiome in the placenta actually is very similar to the profile of the maternal oral microbiome, which I found quite intriguing because it lends plausibility to that link between periodontal disease and preterm delivery. In addition, they found associations with maternal infections, such as UTI, associations of unique microbiome profiles in the placenta, as well as those related to preterm birth. Other evidence for non-sterile fetal environment include the analysis of the first meconium, so the first stool of babies, also revealing microorganisms. And as we all know, the fetus is swimming in this abundant amniotic fluid that they are swallowing and breathing continuously throughout their 9 months of development. So likewise, the initial intubation at birth and evaluating those tracheal aspirates have also revealed microbiome organisms. What we don't know, and things for future study, is what is the role then of the maternal fetal microbiome in fetal development. Ex utero, there are many influences of the microbiome profiles for infants, both in preterm and term newborns. And here is an example of some of them that have been well studied, where when we look at the development of the early microbial colonization of the gut, you want this balance of the bacteria that's present. Diversity and balance is something you'll hear often. And when these modes and birth practices are altered, and also the environmental influences later in infancy, this can disrupt this balance. And some things that we're learning is the delivery mode, vaginal versus cesarean section, is a very important determinant, where vaginal delivery lends itself to a greater colonization for the infant of Bifidobacterium and Lactobacillus, organisms thought to be commensal and beneficial to the host. And this is in contrast to cesarean section, which you'll see more development of Enterococci, Clostridium, and coliforms. The diet is the next probably major determinant in infant microbial colonization, with breast milk being more beneficial, providing those early commensal bacteria of Bifidobacterium and Lactobacillus. It is thought that there's actually these complex sugars and oligosaccharides in breast milk that play a very important role in the epithelial cell and bacterial interactions and being a part of that process of establishing the microbiome. The formula, which is devoid of many of these contents, then leads to a more pathogenic profile in our infants, more of these coliforms and Enterococcus and Clostridium. Hospitalization will lead to colonization of indigenous hospital organisms, often, as we all know, more pathogenic than not. And our medication practices, these are lessons we're learning daily now and over a short period of time of the infant stay in the neonatal intensive care unit, how medications can alter the microbial colonization, especially in the intestinal tract, where receiving prolonged antibiotics or even H2 blockers are changing those profiles and leading to an increased risk of necrotizing enterocolitis, an inflammatory bowel disease in our neonatal intensive care unit with high mortality and high morbidity. And prematurity itself, not only do we see the determinants in a preterm infant, but understanding what happens in early fetal and infancy may actually help us understand what happens later in life. So, for example, with prematurity, there's an underdeveloped glycosylation process. That process that, again, interacts and leads and dictates for that cell and bacteria interaction with one another. So that has allowed a whole new research about glycosylation processes and how that helps in bacterial colonization in disease and health. All of this is important because ultimately the microbiome, or that early microbial colonization in the infant, teaches us about our immune system and helps program our immune system. And the intestinal tract, when fully mature, will be the largest immune organ, 70% of our immune system, and our largest defense barrier. Three main components of this defense barrier are nonspecific intestinal barrier defenses, such as the mucus layer, the epithelial cell layer and tight junctions, and the gut-associated lymphoid tissue, which is the primary site of the activation of the inflammatory response. And the microbiome influences each of these components. It maintains the integrity of that mucosal barrier. It helps augment IgA and the mucus production. It regulates appropriate bacterial colonization. There's a nice competitive force between the different bacteria in regulating that normal homeostatic balance. It activates intestinal immune defenses, and it modulates, therefore, the intestinal inflammation. Ultimately, we as hosts and the receiver of numerous antigen presentations on a daily basis must form a balance between an appropriate tolerance and an inflammatory attack. We must tolerate antigens that are good for our health, such as nutritional components, but we must also be poised to strike an inflammatory attack when faced with a potential pathogen. And an imbalance in the system can lead to intestinal diseases and ultimately even systemic diseases. And as a neonatologist, it's quite humbling that this balance is formed very early in life, and for 10% of the population to 12% who experience a stay in the neonatal intensive care unit, we are ultimately a part of that process and driving what the microbiome ends up looking like, and therefore how is that infant programmed for health and disease later in childhood and in adulthood. An example that we're learning in the preterm infants about these perturbations is the link of an altered microbiome and the development of necrotizing enterocolitis that I referred to earlier. And this is just one example of many emerging studies that evaluates the differences in the microbiome in preterm infants of those who developed and went on to develop necrotizing enterocolitis and those infants that did not. And two things are evident. One is that in these studies, pretty consistently to one another, there's a decreased diversity in the microbiota population. And this was measured in this case by the Shannon Diversity Scale, where infants who went on to develop necrotizing enterocolitis had lower diversity than those infants that did not. The other second theme that we see recurrent in our literature is this, what we call the blooming of the proteobacteria phyla. Where in control infants, there's a relative balance between proteobacteria and the other phyla. But in infants with necrotizing enterocolitis or who go on to develop this, there's that increase, a substantial increase out of proportion to the other phyla of the proteobacteria. But it is important to remember that the microbiome is altered in all of our preterm infants. And continually moving forward, we're seeing literature linking these altered microbiome profiles not only in the intestinal tract but also in the lungs and affecting systemic immunity, such that these associations are linked to the development of necrotizing enterocolitis, as I discussed, but also with chronic lung disease and nosocomial sepsis or late onset sepsis, infections acquired later while they're in the NICU during the NICU stay. And much of these mechanisms are thought to be due to the altered immune ontogeny. These are the first initiators to teach our babies their immune system and the effectiveness of their immune system against these external stimuli and antigens. And when altered, leads to a dysregulated inflammatory response, and together those impair normal organogenesis. You know, in reviewing the content of this talk and what I wanted to share with you, I came across Right, and I apologize. I still don't quite know what happened, but let me try to wrap this up in a couple of minutes to save time for the last presenter and questions. So birth practices that we have now in modern society, the increasing cesarean section rate, the use of perinatal antibiotics, the reduced maternal to fetal, skin to skin immediately after birth, and the variable breastfeeding rates, all of those things that are changing over time are changing that early establishment of microflora. And C-sections are independently linked by the best clinical studies that we can do, where we're establishing these associations to autism, celiac disease, type 1 diabetes, asthma, and obesity. I also encourage you to read a reference in this month's Pediatrics, where they looked at a 2 million birth cohort of term infants and found an increased risk to asthma and arthritis and other immune deficiencies. So where do we go in understanding that early establishment? Well, it's important to protect and restore the microbial diversity. And protect, what I mean by that is to conduct these studies so we can understand how current medical practices alter the microbiome, and then reevaluate those medical practices. The studies on antibiotic use and H2 blocker use has definitely influenced our practices in the neonatal intensive care unit, as we're all becoming more cognizant of what we're doing to the microbiome of the preterm infant and how that lends itself to disease risk. And then the restoration of that microbial diversity. Probiotics has been evaluated in this population, and then I'll briefly mention an interesting study on restoring natural colonization. For probiotics, many of these have been overseas, but it's been difficult to translate to the bedside because, as this meta-analysis shows, different powered studies, different entry criteria, different probiotic cocktails, and different dosages. So we're still somewhat at a loss of what probiotics are going to get the most meaningful effect for us. But also we're learning we need to be cautious. Just like we're manipulating the microbiome on one end iatrogenically through our practices, doing it on the other end thinking we're correcting it, we need to be aware of what these probiotics are doing, especially in vulnerable populations. There's been case reports of septicemia of probiotic organisms for preterm infants receiving probiotics, and a recent case that just came out in the past week investigating the contamination of probiotics for other unknown organisms, and really declaring the need for a pharmaceutical grade quality control product that we can use for preterm infants. And almost to the end, Dr. Bello has this fascinating study. She is from the Human Microbiome program at the New York University School of Medicine. We're trying to, again, restore that natural balance to infants delivered by C-section. And what she's doing in many regions, it's a very large study, only preliminary data are out, by using a cloth and collecting the vaginal flora after the baby's born during normal newborn resuscitation, using that cloth to dry the baby. And preliminary studies have shown that it's beginning to establish a microbiome for these infants closer to the vaginally delivered infants. Not a full correction, but it's a start for us understanding how we can correct our ways and restoring that balance. So finally, returning to those determinants, risks, and therapeutics. Well, the determinants of that early microbiome definitely influenced with increasing evidence by maternal fetal environment, the diet, environment, the other external ex utero environment, and medications. But additional questions we need to understand is, what is the differences between the quantitative and the qualitative changes? What are those specific epithelial host interactions leading to those precise metagenomic and metabolomic effects? And the risks, increasingly, this altered microbiome is associated with risk of disease, especially in that early infancy, preterm, and early infancy term period, and effective through impaired immunity and impaired defenses. For the therapeutics, understanding that protecting, minimizing our iatrogenic reduction in that microbial diversity is important, as well as restoring strategies to return to that natural homeostasis, but also a bit of caution and being aware of the potential dangers with that manipulation, especially in our vulnerable patients. Thank you, and thank you for letting me return.
So let me introduce Dr. Floyd Dewhurst. He's a professor of oral medicine and infection immunity at Forsyth Institute and Harvard School of Dental Medicine. His background has been the work on the oral microbiome before the term, deciphering the oral microbiome before the term microbiome was actually created. Dr. Dewhurst's primary research has focused been on defining the diversity, genetic capability, and pathogenic potential of microorganisms present in the human oral cavity. He has used sequencing of the 16S ribosome RNA genes directly from cultivated oral bacteria, and also sequencing of clone genes derived from oral plaque DNA to identify all the bacteria that can actually survive and live in our oral cavities, both cultivated and previously uncultivated. And the results has identified approximately over 700 species or phylotypes of human oral bacteria that are described in the human oral microbiome database. For many of the strains that at the Forsyth team has put together, has identified, they've actually made them available to the scientific community for previously unnamed and uncultured species. So in addition, along with colleagues at the Department of Microbiology, he has led the way in deciphering the complex oral microbiome, and today 65% of the species in the database can be grown. And apparently in his next phase of research, Dr. Dewhurst is working on developing new innovative methods to cultivate the remaining uncultivated species. And his ultimate goal is to have a full genomic information for all species that comprises the oral microbiome. So we have a really, really wonderful presentation by Dr. Dewhurst on the human oral microbiome, and his abstract is actually on the website, so I'm not going to read it. And please continue to submit your questions until the end of all the presentations. And now I'll hand it to our speaker, Dr. Floyd Dewhurst. Please enjoy.
Okay, can you see my slides?
Yes, we can.
Hello, can you see my slides?
Yes, we can see your slides.
Yes, I can see your slides. Yes.
Oh, okay.
Yes, we can see you.
Okay, great. Okay, so I'd like to tell you about the oral microbiome. And the oral microbiome has actually been studied for the last 340 years. Anton von Leeuwenhoek took a toothpick and scraped between his teeth and looked at it under his first crude microscope. And he was able to see leptotrichia and spirochetes and streptococci and selenomonases swimming very rapidly. And thus began the very beginnings of oral microbiology. In the 1850s through 1980s, cultival microbiology was the main way that we understood what was in the mouth. Investigators such as the Moores at Virginia Polytechnic Institute and Sokransky at Forsyth, as well as many other work to isolate and name over 200 organisms from the human oral cavity. 16S ribosomal RNA was first used to study the cultival Campylobacters in 1988. And just 6 years later, individuals started making clone libraries to figure out what the uncultival organisms were, such as looking at the oral treponemes by Choi et al. Finally, the diversity of what was in the subgingival and then the oral plaque started coming out around 2000, and Dave Relman in his paper in PNAS and Bruce Pastor and myself in J-Bacteriology started figuring out what comprised the oral microbiome. However, there was a major problem in 2004 that was really impeding oral microbiome research, and that's that there was no naming scheme for the 16S RNA phylotypes. That one individual would get a clone and somebody else would get one is for the same organism, but they'd each have different naming schemes, and there was no way to know what the organisms were. Another thing is that there was at that time, there were about 7 or 8 genomes for oral microbiorganisms, and that was it. everything else that was known genetically about all of the other 700 organisms was their 16S ribosomal RNA sequence, and that's pretty meager. Also, there weren't isolates available at culture collections except for the named organisms. So the National Institute of Dental and Craniofacial Research in 2004 put out an RFA to understand the metagenome of the oral microbiome. And this was actually ahead of the human microbiome by about 5 years. And one of the things that I thought would be important is to establish a foundation for the oral microbiome and metagenome where I would create a taxonomic framework for unnamed organisms, have reference genomes for a wide diversity of organisms, up to 100, and also seek to get isolates of previously uncultivated organisms and make them available. In doing that, I created the Human Oral Microbiome Database, and it describes approximately 700 of the most prevalent endogenous organisms in the mouth. And it has a taxonomic scheme, it has page descriptions of each organism, it has blast facilities such that you can take 1,000 concatenated 16S sequences from clones, drop them on this page, and get back identifications that go back to these to this curated database with defined taxa. It also contains the genomic information on the organisms so that you can get down to the gene level and look at the amino acid or nucleic acid sequence of any particular gene. The diversity of the oral microbiome is quite broad. We know of at least 13 different phyla that comprise the oral microbiome. The most species are in the Firmicutes, and then Bacteroides, Proteobacteria, Actinobacteria, Spirochetes, Fusobacteria, and then some lesser-known organisms such as TM7, GNO2, SR1, and Chloroflexi. So a very broad diversity in the oral cavity. Interestingly, at this point, we've already named 50% of the organisms that are present. We have isolates for another 100 or 15%, and those that are known only from cloning studies comprise about 35%. So in many environments, only a very small percentage, sometimes even less than 1% of the organisms in an environment have been cultivated. But again, in the oral cavity, we've cultivated 65% of them and are working on the last 35% now. Also, we have over 1,300 complete genomes on these organisms, representing 368 of the nearly 700 taxa, so that more than half of all the taxa now have genomes, and of those that we can cultivate, 83% have genomes. And the utility of these genomes is for studying the pathogenesis of the organisms, but also the genomes gives you the proteome so that you know the amino acid sequence of each protein that the organisms can make, and then you can do, using mass spec techniques and so forth, do proteomics. You can do transcriptomics, again mapping things back to these completed genomes, and looking at the pathways based on the genomes, one can figure out what metabolites should be made by each of the different organisms, which help metabolomic studies. So all of this information is available at www.homd.org, and it's described in a 2010 paper. The biogeography of the oral microbiome is quite complex. There are actually at least 10 or more niches in the oral cavity, and interestingly, each has a microbiome that can be differentiated from the other, all of these different sites. So while your tongue normally sits up against the roof of your mouth, unless it's been chopped open, as here, separated by a thin coat of saliva, if you were to take a Q-tip and rub it either on the roof of your mouth or your tongue and do an analysis of the organisms present, with 100% certainty you could tell whether it was from the roof of the mouth or the tongue, that these microbiomes are unique. Some of the differences were first described by S in 2005. The oral microbiome is really important for health and disease. The most common infections are tooth decay or caries, periodontal disease. These two affect almost all individuals to some degree. And then other diseases such as strep throat, tonsillitis, sinus infections, middle ear infections. Middle ear infections are the most common cause for children to visit physicians and the most common reason for antibiotics to be prescribed. Pneumonias, often the organisms pass to the lungs via the oral cavity. The organisms from the mouth are also important in systemic infections, obviously streps and bacterial endocarditis. Organisms from the mouth can get into the bloodstream, causing strokes, preterm birth or low birth weight, and infections or abscesses in all kinds of distant locations such as brain, liver, or bone. One of the new areas that I think you'll find interesting is that salivary diagnostics are coming into play. And obviously looking at the organisms present in saliva, there are tests for caries, periodontal disease, also tests for viral agents such as HIV, hepatitis, dengue, herpes, Epstein-Barr virus, even, and many other diseases. The salivary diagnostics have been reviewed in the two articles listed here. A lot of salivary diagnostics look at host molecules, but as cited here, some of the salivary diagnostics are also using the microbiome as a component of their diagnostics. A final area that I just want to touch on is prebiotics, probiotics, and replacement therapy. Obviously, one of the things that we know the most about is if you want to stop dental decay, one needs to reduce the intake of sugar. So without actively putting in different bugs, if you put in a bunch of sugar into your mouth, you will eventually select for organisms that are resistant to acid and make acid, and you'll end up with a very caries-prone situation. Streptococcus mutans is the organism that is most responsible for causing caries, and in the first replacement therapy that was worked out more than 15 years ago, Jeff Hillman created a strep mutans that rather than making lactic acid, it made ethanol, and that makes the organism no longer cariogenic. And therapy with this organism is being pursued by orogenics. Another example is many people are afraid of eating gluten. Gluten is only partially broken down by the normal digestive enzymes and leaves fragments that are incredibly immunogenic. It turns out that Rothia area produces a protease that breaks down these residual immunogenic peptides and might be useful eventually for treating gluten sensitivity and so forth. Eva Helmerhorst is working on that. Finally, probably you all have heard of microbial transplants for things like C. difficile infections in the gut. There was an interesting paper that just came out about a week ago on transferring oral organisms, where they found that French kissing can transfer up to 80 million bacteria, and that people that one kisses regularly have much more similar oral microbiomes to one another than they do to random people in the population. It isn't surprising, it's been known for a long time that children get their oral microbiome primarily from their mother or principal caregiver. So in conclusion, the oral microbiome is an area that is easy to study. It's easy for access and sampling that can be used for diagnosis. The majority of the organisms are cultivated and can be worked with. Strains are available from culture collections, and the genomes for the majority of the bacteria are available. So I think that that's it. Thank you.
Thank you, Dr. Dewhurst.
Well, thank you.
For such a very informative and concise presentation. I definitely now will not do any more French kissing.
I don't think that was the message. That's not what I took away.
So in reference to the presentations by Dr. Martin and Dr. Dewhurst, you have a flavor of the basic concepts of the human microbiome as it relates to human health and disease. We know that the microbiota is essential for maintaining health, as described, and also is altered in disease. And the composition and function of the healthy gut and oral microbiota remains to be clearly defined. And this leads a nice segue into Dr. Sasha Vlasov's presentation in reference to technical resources. So today we're really excited to have Dr. Sasha Vlasov, a developer and application scientist and specialist with a vision, who is actually currently at Thermo Fisher developing and providing tools to facilitate your microbiome research efforts. Without going into details, his bio is actually on the website, so I'm going to go right into his presentation. He will describe workflows and the latest tools for human microbiome research, including sample collection, storage, isolation of microbiome, DNA and RNA sequencing, and subsequent data analysis. I'll hand it over now to Dr. Vlasov. Please enjoy.
Thank you very much, Vincent. I will give a high-level overview of the field of microbiome research and next talk about the tools typically used for studies of microbial communities. Can we go to the next slide, please?
Such as just click in the slide window and you should be able to do it there.
Okay, good, it worked. So from the two previous fantastic presentations by Kemi and Floyd, you already learned that microbiome research is an amazing and exciting new area growing exponentially these days. Microbes associated with human bacteria include not only bacteria, but also eukaryotes, archaea, and other types. Bacteria that are present on the human body in substantial amounts, and they outnumber actually human cells by up to 10x. And viruses and other microorganisms are substantially less studied, but they also represent very diverse and huge community. Human body typically contains 2 to 5 pounds of bacteria. That translates into 1 to 3% of our body mass. That is a huge number, and it's amazing that it was gone unnoticed for so long. There are about 1,000 different species of bacteria populating different parts of our body. And these are only bacteria. Once again, I wanted to emphasize that bacteriophages and fungi are substantially less studied because there are no good tools to do that, and it's very challenging and expensive type of analysis. Normal human microbiota has many functions and beneficial effects to human body, which are only partially understood at the moment. We know that they perform carbohydrate fermentation absorption. They have very important roles in immunity. They secrete substantial amounts of vitamin K and B12, for example, stimulate development of certain tissues, but there are many, many other important functions yet to be discovered. And it's very important to keep in mind that none of these effects stay local. So, for example, all the substances secreted by bacteria reside within our guts. They don't stay local, but they travel throughout the human body and have very distal effects on brain, lungs, etc. So overall, bacterial ecosystem is essentially a new organ dramatically influencing human health. It's a very sophisticated new organ which we don't really understand at the moment well, and it's extremely important to invest more in this kind of studies. So overall, there are several microbiome initiatives. Probably the most famous one is Human Microbiome Project that was started by NIH back in 2008. Since then, it was about $200 million investment in 80 research institutions worldwide. And the primary goal was to understand who is out there, to characterize microbial communities residing within different parts of human body. To accomplish that, 240 healthy subjects were involved, and swabs were taken from 15 to 18 different sites of the body, and the microbial communities were analyzed by next-gen sequencing. Very nice reports we put together. They're publicly available, and now we have really good understanding of bacterial communities from all body sites. One should keep in mind that this is just phase 1 of the project. Now NIH initiated phase 2 of this program that is starting to look at whether particular microbiome compositions correlate with certain diseases, disease risks, and responses to drugs or other treatments. And of course, another big question, in addition to trying to understand who is out there, we need to ask this question, what they're doing. What are the functions for this amazing bacteria residing within the human body? In addition to NIH Human Microbiome Project, there are many other initiatives. For example, American Gut Project that is also huge and it has very similar goals. And all this research is published a lot. For example, there are several journals that emerged in the last 2 to 3 years that are solely focusing on human microbes. And if you go to PubMed, there are thousands of publications available. For example, this year, about 4,000 papers were published on the microbiome subject, and the number of publications is growing annually about 20%. And of course, we shouldn't forget that human body, despite it has substantial variety and amount of microbes, there are substantially more microbes around us. And I'm glad to see that there's Earth Microbiome Project exploring bacteria, fungi, and viruses in soil, in plants, in water, and they're trying to understand how we influence that and how Earth Microbiome is influencing humans. In addition to basic research, of course, many people are exploring the practical applications. And I'm talking primarily about diagnostics and therapeutics. It is very obvious and very clear that all our knowledge about human microbiome can be immediately translated into innovative therapies and diagnostics. And a good number of companies are exploring that, ranging from very small startup companies through gigantic pharma such as GlaxoSmithKline, Bayer, Johnson & Johnson, etc. To give you just a couple of brief examples, one company is exploring the potential of development of tests for premature labor based on analysis of vaginal microbiome. And for therapeutics, probably the best example is fecal microbiota transplants that became really popular option this day, and it enables to treat patients with recurrent C. difficile infection extremely efficiently. So with single administration, you cure up to 95% of people. And these are just a couple of quick examples. There are way more strategies how you can translate the data from microbiome projects into clinics. It's really, really exciting. Human microbiome projects are very diverse, and there are many, many questions that scientists are asking. In order to make substantial progress forward, it is extremely important to have the right tools for this kind of research. And there are many, many challenges. To give you an idea, for example, many of these microbes cannot be cultivated. So, for example, talking about GI tract, it's huge diversity of microbes, and very, very few of them can be cultured in the lab. Another problem is that there are so many of microbes of all kinds, and they can be viewed as very sophisticated ecosystem, and it's extremely challenging to study them one at a time. So just you need to understand all the networks and pathways and their interaction between them. So you should come up with sophisticated systems, artificial systems, how to study smaller and simpler communities to model the real system. So this current slide shows just one of the examples of workflows, a very simple one enabling endpoint analysis of microbes by sequencing. This is a typical workflow for human microbiome project. It all starts from sample collection that is typically either stool samples to explore GI tract communities or swabs from different types of body, and primarily oral cavity, nasal cavity, skin, etc. Once the samples are collected, next they can be preserved by freezing or they can be processed fresh at room temperature. But in some cases, when scientists are collecting environmental samples in the field, they choose to use chemical reagents to stabilize the sample and store it at room temperature. Once samples are ready for analysis, you can move forward with the recovery of RNA and DNA using a variety of kits that I'll be talking about in a minute. And the final stage is endpoint analysis by next generation sequencing, or if you're studying smaller communities of known bacteria, of course, you can use qPCR with Tachman assays. So speaking about sample preservation, the most popular reagent is RNA-Later. This reagent was developed originally for stabilization of RNA in fresh tissue and cells, but it can be also successfully used for stool and swab samples, and also it's doing a pretty good job at providing extra stability for DNA. Clearly, DNA is much more stable analyte versus RNA, but extra stability never hurts. Workflow is very simple, as you can see from this slide. You collect the sample, add RNA-Later reagent, it penetrates tissue of interest and preserves RNA or DNA. Then when you're ready for analysis, you remove excess of reagent and move forward with sample homogenization and recovery of nucleic acid content. When you move to next step, the choice of technology will be dictated primarily by your throughput and specific needs. If you want to isolate RNA or DNA from very few samples and you perform this procedure, let's say, once a week or once a month, organic extraction is one of the best options because it's an extremely simple protocol and these reagents are cost efficient. It's a very, very cheap option. When you're processing larger number of samples, let's say in the range from 6 to 24 columns are extremely popular, and this can be done by centrifugation, or you can use vacuum manifold to process larger sample volumes through columns. One should keep in mind that columns typically provide higher degree of purity for your analyte versus organic extraction. And when your project requires processing substantial number of samples, 50 plus or hundreds of samples, of course, magnetic beads are superior to earlier discussed options. They allow you to easily automate the process, and you can recover RNA or DNA from hundreds of samples per day if you use appropriate instrumentation. So I'll just briefly highlight all of these reagents one at a time. Trizol is probably the best example of organic extraction reagents. It has years worth of history. It was successfully used for many different sample types. It has amazing lysis capacity, flexible formulations for difficult samples, scalable, it's single step, very simple workflow. It allows you to isolate not only RNA, but also DNA and protein from the same sample. And it's also available with complementary max bacterial enhancement reagent for challenging samples. Column-based kits are extremely popular in the field, and there are many companies offering that. Here I just wanted to highlight challenges specific to microbiome research. The first challenge with your sample is robust and uniform lysis. You definitely want to make sure that whatever technology you choose allows to robustly lyse all microbes in a sample. Otherwise, you'll never see them, and it can completely change the course of your project. So you simply won't notice some of the bacterial species that are resistant to lysis. The two solutions, first of all, you can use mechanical disruption with the beads, and bead beating is widely used by scientists worldwide. Another option, you can use carefully optimized enzymatic or chemical digest. That's another elegant strategy. Second challenge is efficient removal of PCI inhibitors, and this is especially the problem when you're dealing with stool and soil samples. that have many molecules that co-purify with RNA and DNA and negatively influence your downstream analysis. Two typical solutions. First of all, we can introduce additional washing or precipitation procedures to remove them. And another elegant solution, you can use robust enzymes for downstream analysis. So that means that you can use any standard kit to purify your DNA or RNA, and it will contain some inhibitors, but you can compensate for that by using mutant polymerases downstream that are capable of PCR or RT applications in the presence of very high levels of inhibitors. And the final product is magnetic bead-based kits. MadMax is a really good product that stands out. It allows to accomplish maximal purity for samples. The protocols are very simple, and the process can be completely automated using a couple of instruments shown here on the right. And you can manipulate either with 96-well plates for high throughput, or for lower throughput and larger volumes, you can choose 24-well plates. So to summarize this very brief overview of kit and reagent options, microbiome research is a new hot topic, and fundamental aspects are extremely important. We definitely want to understand more about who is out there and what are the networks and pathways of all these microbes. Knowledge of human microbiome can be readily translated into diagnostics and therapeutics, and a large number of companies are making steady progress with that. Some of the drug candidates are already in phase 2 and 3 clinical trials, and fecal microbiota transplants are already performed worldwide for the last few years. There are many clinics that do that routinely. Third, we developed a set of reagents and instruments enabling isolation and downstream analysis of microbial DNA and RNA, and novel kits from Invitrogen and Ebion will be launched shortly. With that, I wanted to say thank you, and please let me know if you have any questions.
Well, thank you, Sasha. We really appreciate that. We are almost at the end of our time, but we're going to continue on for a few minutes because we have some great questions. So we appreciate, folks, if you need to leave, but if you can stay, we would be very happy to answer some questions. I just want to also note that this is just a taste today. We're doing a whole series on the microbiome, a short course, in April, which are 4 Thursdays in a row at 11 a.m. Eastern. That's Eastern U.S. That's April 9th, 16th, 23rd, and 30th. You'll be receiving more information about that. I'm going to share the first question, and I will also say we didn't get a chance to really tell you about Dr. Kuo, but you can read his bio on the website as well. Here is a question from Jason Kralik. Great content. For Floyd, is there evidence for oral organisms or byproducts entering the brain, crossing the blood-brain barrier, and having, and I can't read the rest of it, probably having an effect for some reason. I can't read the rest of this question. But yeah, could you talk about the blood-brain barrier, please?
Yes, definitely. Organisms from around the teeth get into the bloodstream, and from there get to the brain. And people have been able to find particularly treponemes that have crossed into the brain. And there is, a small amount of work suggesting that oral bacteria, particularly the spirochetes, could be a contributing factor to the inflammation that contributes to Alzheimer's. So there is evidence for that.
Does anybody else want to comment on that question? Is that relevant to your thoughts, Cami? Dr. Martin?
I don't have any additional. I don't think we have much in the way of evidence right now for the term and preterm infant. I'm only pausing because one question for us is that we know some of these diseases, such as necrotizing enterocolitis, are independently linked, even though we think they're intestinal driven, independently linked to neurocognitive deficits. So is that because of the immune and inflammatory balance of the microbiome, or is it a secondary dysregulation of the inflammatory response and cytokinemia? And I think a lot of those are trying to be explored, but for the vulnerable kind of preterm with an underdeveloped blood-brain barrier, there does seem to be important linkage in those neurocognitive deficits to our neonatal morbidities, and we don't fully understand why.
Yeah, one of the things is that the organisms from the oral cavity, whether contributing to cardiovascular disease or anything else, the organisms sometimes do not have to get to an area alive. They are full of, if they are phagocytized by a macrophage or something like that, the phagocytized organism can be then translocate with the macrophage to anywhere else in the body. And some of these lipids are incredibly potent at stimulating the toll system, creating cells that create fantastic amounts of IL-17 and so forth. So we actually know much more certainly that specific lipid products from oral microbes get to distant sites, and more just suggestoid data that the organisms themselves get there. But their products absolutely get there.
Thank you. We have a question from Naya Adaluti. I hope I'm saying that reasonably well. It was found lately that most testing kits and other reagents for analyzing DNA contain strain microbial DNA that can overpower the signal of the native bacteria. Does your company, and this is obviously for Dr. Vlasov, does your company guarantee materials are sterile?
That's a great question. I saw this publication recently, and this seemed to be the problem for ultra low input. So when you're dealing with extremely small amounts of material, indeed, some of the kits contain tiny amounts of DNA and RNA that can negatively impact your studies. But in typical microbiome research, when you're dealing with swabs or stool, the amount of bacterial DNA is orders of magnitude larger compared to these impurities. So this seems to be a problem for a small community of scientists, and we want to address that. We plan to run additional tests for our kits and confirm what are the levels of this contamination, whether it's something of a problem or not. If you find it to be a problem, then we'll need to launch ultra-pure kits for special applications.
Yeah, this is particularly important for if we know that when people chew that they get little bacteremias, and 99.99% of the time these bacteria are cleared by the endoreticular system. But if one wants to try and see what bugs are circulating where there are just a few organisms per mil in blood, then this is where that type of contamination becomes a tremendous problem. And what sort of unwary researchers have found, looking at samples with none to just a few individual cells, is that they get the standard things that are in drinking water and that you would expect in reagents.
Thank you.
That's a good point.
So we have a question from Tom Fair, who is actually our Director for Strategic Alliances here at the BRC. I have read that the microbiome is quite dynamic during the day-night cycle. Does the oral microbiome have a true equilibrium state?
I guess that's directed at me. I'm not sure that I can answer that. I mean, it is my understanding that the oral microbiome does vary with circadian rhythms and so forth. I think the question of what is the stability of the oral microbiome in an individual over time, it's actually quite remarkably stable in that many of the same organisms that if you get the organism out and fingerprint it, that it is the exact same strain will persist for a year or more, and that if you look at somebody's organisms and then you look a year later, they're much, much more similar to themselves than to anybody else. And so there is that stability. But getting a sample, the numbers, if you get a clump of some organism when you're sampling, the relative numbers of different strains will certainly go up and down from sample to sample.
Well, thank you. We're going to have one more question, and then we'll ask Dr. Kuo to pull everything together for us. And I appreciate almost everyone has remained with us. Thank you. And I'll just, I'm going to put up a slide in a moment with our website on it, and you can learn all kinds of great things from there. And all of the ideas for all of the programs that we develop come from people who participate in them. So if you have an interest in helping us to develop a program, or there's one that you'd particularly like to see, you can reach me through the website, and I'd be very happy to hear your ideas and see what we can do together. We're a small organization, which means we're very agile. So our final question from those listening is, what role does breast milk play in shaping an infant's microbiome? And what studies have been done to compare breastfed and formula-fed infants along these lines?
Yes, so the there's a fair amount of literature out there. There's several studies looking at breastfed babies compared to formula-fed babies. As I mentioned briefly, there's probably a lot of factors within breast milk. I think when someone did a proteomic analysis of what's in breast milk, it was over 500 to 800 different proteins and other immunomodulatory agents that we can't replicate in formula. And one of the key things, again, is the oligosaccharides. A lab of Lars Bode is really doing some excellent work in that, and definitely worth looking up his literature. And the oligosaccharides are those complex sugars and glycans that are important in that cell, epithelial cell, and bacterial interaction. So definitely some plausibility and biological plausibility for the differences we see between those two populations.
Terrific. Well, Dr. Kuo, would you like to share your thoughts as we conclude? And actually,
Sure. Yeah.
Thank you.
Sure. Yeah, so first I want to thank Howard for initiating and organizing with the Biopharma Research Council this wonderful webinar. Joanne, her team, and our presenters, Cami, Floyd, and Sasha. As we heard from Dr. Martin and Dr. Dewhurst, the presentations were very high level overview in terms of the human microbiome as relates to human health and disease, from Cami's talk in terms of microbiome and their organisms as relates to labor and risk of premature delivery. And you heard a lot about in terms of the number of microbes in the oral cavity as well. And also Sasha's presentation provided a framework and technical tools to be able to further along your research in terms of the human microbiome. So also, although the benefits of the microbes are not known to science, the human microbiome's importance and in some cases necessity to human health is a notion that is taking center stage and is generating a lot of questions. And recently, a lot of studies in the microbiome has mushroomed as researchers explore what makes up the huge populations of microbes in and on our bodies, exactly what they're doing and how they can be manipulated to promote health and combat disease. As you've seen today's presentations, and there's many studies and different approaches are definitely now underway to unravel these relationships between the microbiome and diseases ranging from, as you've heard today, from autism to cancer to development. The possibilities are rising from the view of the microbiome as working with human cells and physiological pathways and potentially fighting disease has triggered a huge demand for research and technologies like next generation sequencing, sample prep, as you heard from Sasha's presentation, that can open windows to our basic understandings. Yet there are still key questions pertaining to them that are sort of important, whether alterations in the microbiota are causative or merely association with disease, whether therapeutically manipulating the microbiota by dietary changes or even or restorative therapies or immune modulations would alter the disease course. So this was essentially today was to sort of present a higher overview in terms of different aspects of the microbiome, and also the tools that can enable further down the research. And I really appreciate, again, the Biopharma Research Council, Joanne and her team, and our presenters today, and also thank Howard for initiating this topic.
Thank you, everyone, and we'll look forward to seeing everyone throughout the year, and we'd love to hear from you. Take care. Go ahead.
And the presentation will also be online afterwards, so you can always go back if you missed certain sections. You can always go back to the website